This Week in Virology, the podcast about viruses, the kind that make you sick.
This Week in Virology, episode 145, recorded on August 12th, 2011. Hi everybody, I'm Alan Dove, and it's time for TWiV, the podcast all about viruses. Joining me this week from North Central Florida is Rich Condit.
Hi, Alan. How you doing?
I'm okay. How about you, Rich?
Good. What do you got for weather today?
Oh,
we got through the weather report.
It is glorious here.
Oh, that's good.
It's one of the 5 days a year that Massachusetts just gets beautiful, beautiful weather. So it's about 80 degrees and clear skies and just
Excellent.
Lovely. How about you?
Excellent. 93. I'm used to it. It's good. It's not too humid. It's okay. It would kill most people, but it's good for me.
I guess you've adapted to it.
Yeah. Something's wrong here, Alan.
Yeah, it seems a little different.
Is someone missing?
I guess Dixon couldn't make it, could he?
Oh, no, Dixon's not here.
Yeah.
How about Vincent?
Oh, right.
Where's Vincent?
He's not around, is he?
Darn, jeez.
Gosh, our listeners probably picked up on that.
Well, you think we can handle it?
Well, let's give it a shot.
Okay.
Okay, so here we go, the invincible TWiV.
Excellent. Vincent, by the way, is off traipsing around Europe with his family. He deserves a break.
Yes, I think he does.
We thought we'd pick up a little slack for him.
Yeah, it's about time we did a little work.
Yeah, he said he thought he could cut us loose. We'll find out whether that was a good idea or not.
He'll rue the day.
Right.
All right. So yeah, this first thing we've got is actually really, really interesting. This was you picked this out, and
actually, before we even get to it, I want to fix the immunology bug.
Yes.
Episode.
Yes.
So we were last episode. We discussed a paper where they, as a potential HIV therapy, this was in a model system, they isolated T lymphocytes from, wow, I'm blanking on it. Was this entirely mice?
I think it was mice, yes.
Yes. Isolated T lymphocytes, knocked out CXCR4, and then, no. There was humans, okay? And they used a humanized mouse in the end. They isolated human T lymphocytes, knocked out CXCR4, which is an HIV co-receptor, and then stuck the lymphocytes back in the animals and looked to see whether they were infectable by a virus that required that as a co-receptor. And it was all looking pretty good. And we had this discussion. The problem is CXCR4 is a gene that is required during lymphocyte development and also several other aspects of development. And so the idea is, but it may not be required at the late stages of T cell development. So the idea was that if you wanted to engineer this, you would have to engraft T cells, and we had a question as to whether you could do that successfully and expect that graft to maintain itself for a long period of time.
Or would you need to transplant some kind of stem cells in?
Right. And so I checked with my immunology friends, and they said, no, you can graft T cells, and they will last for a significant period of time. That the person I talked to was unwilling to say lifetime, but for a very, very long period of time. So they will regenerate themselves. These are naive T cells, right? So that's okay.
Right.
Now, whether CXCR4 is going to actually affect their function down the road is another question, because in T cells it's a chemokine receptor, and so it could influence how those cells home to different sites and that kind of stuff. So there could be other sorts of problems. But the basic question as to whether or not you can effectively permanently engraft T cells, the answer to that is yes.
Right.
All right?
Yes, and after the show, I did a few quick Google searches, and I kind of got a little bit of similar information. I said, oh, yeah, we screwed that up. So apologies to the audience.
That's okay. There are probably some immunologists out there who were cringing, but that's okay. We're mortal, right?
Yes, we are. We are, believe it or not.
and we're still learning. I'm still in school.
Oh, absolutely. Well, I learn something new on every TWiV.
Yep, it's great. All right.
So, including this paper that you wanted to talk about, which I am a little embarrassed to say this historical aspect of virology, which is pretty important, was completely news to me.
Well, I'm glad we could come up with something new.
Yeah.
So, a little So this is a history lesson, and I will start out with a little personal history. I think I've said several times before on this show that, I've over my career, I probably shouldn't admit this to all of the young and young listeners out there, but over the history of my career, keeping up with the literature has not been my strong suit. That's TWiV is really good for me in that way. But occasionally I go off on these reading benders, and one of them was while I was writing my thesis. I think it was mostly a procrastinatory thing. Okay. And I dug way back. I was working on bacteriophage, and I wound up in the stacks one day, digging way back to the very beginning. I had been aware of this, but I wanted to look at the paper that described the first literature description of bacteriophage, which is a paper by Frederick Twort called An Investigation on the Nature of Ultramicroscopic Viruses, published in The Lancet, which is sort of, would you agree that it's sort of the British equivalent of the Journal of the American Medical Association?
Yes, that's a good analogy.
Premier medical journal from Britain with a long and glorious history. So this is from 1915, volume 186, pages 1241 to 1243. It is not open access.
Right.
But if you have a library that keeps all the old volumes as we do, you can go check it out. And in this paper, which is as interesting for the way it's written,
yes,
as anything else, in this paper, he describes what we now know are bacteriophage. So I'd like to put this in a little bit of context with some history. So this is 1915. At that time, germ theory was well established. That was from 1860 to 1880. Pasteur and Koch sort of refined and consolidated germ theory. And in fact, viruses, both plant and animal viruses, had been described. So the earliest description of a virus was by Ivanovsky in 1892, where he described tobacco mosaic virus, and at this point, they were defined as things that won't pass through that will pass through a porcelain filter,
right,
which will filter out any bacteria.
Filterable agents.
Filterable agents. Bayerink sort of rediscovered TMV in 1898. Loeffler and Frosch in the same year showed that foot and mouth disease was caused by a filterable agent. 1908, Ellerman and Bang described a filterable agent that could cause leukemia in chickens. And a famous paper that I'd like to do on another occasion, I've read as well. In 1911, Rouse described a chicken sarcoma virus.
Rouse sarcoma virus, yeah, that's an important one.
Right. So by the time Twort comes around, 1915, the notion of a virus was, at least in plants and animals, as a filterable agent, and also as something that required, apparently, up until that time, required the presence of living tissue to grow on, to propagate. That was well established, but the actual nature of these things was not understood. And what I have trouble with in reading this paper, among other things, is putting my head in the space that these guys were in, because I know too much. Right?
It's often hard to read the really old literature because they're talking about stuff and you're thinking, wait, what is this talking about? It actually, when I read the Twort paper when you sent it to me, I was thinking, wait, is he talking about plaques?
Right.
But of course he didn't know what they were.
Right. What's he thinking?
Right.
Right. So this paper, as we've said, it's as interesting for how it's written as anything else. It's so Lancet is a sort of a standard size format journal with 2 columns, and the whole paper is, if you add it all up, 2 pages. So 4 columns.
And one author talking about just some stuff that he saw.
Yeah, there's no tables, no figures, no nothing. It's just a narrative.
Which
it's
is
like
which
this
is
guy's
also
talking to you over lunch.
Which is also typical of the really old literature. It was not only was it expensive to publish figures, but in most cases they didn't have good pictures of any of this stuff, so they would just talk about a series of observations.
And by the way, this guy is the director of an institute called the Brown Institute, which is basically a veterinary hospital. And on the side, he has a little bit of money to play with stuff, and he's mostly a bacteriologist. His real claim to fame is that he discovered vitamin K sometime before this. So at any rate, the way he sets this up is that he is trying to cultivate viruses as free agents, really. And this is what kind of blew my mind, because I thought, well, wasn't it established at this point that you needed living tissue to grow these things on? And I guess maybe it wasn't really firmly established, and that he's thinking that maybe you should be able to find the right media to grow these things by themselves in the absence of anything else.
Right, and in fact, he talks about trying a whole bunch of different media. Right?
Yeah, he says, as a matter of fact, what I'd like to do is actually read some of this stuff to get the real flavor of the paper. It's not real long. So he says, attempts to cultivate these, that is viruses, from such materials as soil, dung, grass, hay, straw, and water from ponds were made on specially prepared media. Several hundred media were tested. It's impossible to describe all these in detail, but generally, agar, egg, or serum was used as a basis, and to these varying quantities of certain chemicals or extracts of fungi, seeds, and etc. were added. The material to be tested for viruses was covered with water and incubated at 30 degrees C or over for varying periods of time, then passed through a Berkfield filter. That's a diatomaceous earth filter, which I think functions essentially as a porcelain filter. And the filtrate inoculated onto the different media. In these experiments, a few ordinary bacteria, especially sporing types, were often found to pass through the filter, but in no case was it possible to obtain a growth of a true filter-passing virus. So I guess he's like looking for a colony or something.
Yeah,
right?
he appears to be, and this type of approach was, again, fairly typical. This idea of high-throughput screening was not invented by drug developers in the 1990s, contrary to what some people might believe. A lot of science was just throw stuff at the problem.
Yeah, it just occurred to me, this is virus discovery, 95 years old,
yeah.
right?
Yeah, this is how you went looking for viruses a century ago, or almost a century ago, was apparently you just mixed up whatever you thought they might be able to grow on, and you try to grow them. And I guess he's expecting some kind of colony to arise out of
guess so. So at any rate, none of that worked. And so he decided that he would, he said, he tried, one of his sources of material was ironically, vaccinia.
Right.
So this is the smallpox vaccine. And he got this because it was readily available and fairly well characterized. And interestingly, everybody knew, this is amazing. We talked about this before. That stuff is calf lymph, right? It's grown on the bellies of calves and harvested. And if you streak it out, it's full of bacteria,
right,
of all different kinds. Would never pass muster with the FDA nowadays. No way. So they knew that, and he seems to be thinking that maybe the bacteria in this thing might be important for growing the virus or something like that. So he, as a source of virus, he played with this, and he said, So here's the basic discovery. Inoculated agar tubes after 24 hours at 37 degrees often showed watery-looking areas, and in cultures that grew micrococci, it was found that some of these colonies could not be subcultured, but if kept, they became glassy and transparent. So I guess on the dish he's seeing, watery-looking areas, and if he looks close at them, there's no bugs, it's just granules. And he also finds that he can grow colonies, but they will become glassy and transparent.
Right.
Let's see. A further experiment showed that if a colony of white micrococcus that had started to become transparent was plated out instead of being subcultured as a streak, then the micrococci grew and a new streak of pure culture of these colonies could be obtained. So he can pick bacteria out of these things that are becoming transparent and streak them out and get colonies that are free of this problem.
Right.
Okay? So he can cure them.
Well, not cure them, he can save them.
Right. On the other hand, if the plate cultures were left, the colonies, especially in the first dilution, soon started to turn transparent, and the micrococci were replaced by fine granules. This action, unlike an ordinary degenerative process, started from the edge of the colonies, and further experiments show that when a pure culture of the white or yellow micrococcus, isolated from vaccinia, is touched with a small portion of the glassy colonies, the growth at the point of the touch soon starts to become transparent or glassy, and this gradually spreads over the whole growth, sometimes killing out all of the micrococci and replacing with fine granules.
So he's found a transmissible agent.
Exactly. He can take a bit of this glassy stuff and touch it to a colony that's okay, and it will transform that colony into a glassy thing.
Right.
The transparent material, when diluted 1 in a million with water or saline, was found to pass through the finest porcelain filters with ease, and 1 drop pipetted over an agar tube was sufficient to make that tube unsuitable for growth of the micrococcus. And then he describes basically plaques.
Yeah.
That is, if the micrococcus was inoculated down the tube as a streak, this would start to grow, but would soon become dotted with transparent points, which would rapidly extend over the whole growth. Plaques!
Yes.
All right. The number of points from which this starts depends on the dilution of the transparent material, and in some cases is so active that the growth is stopped and turned transparent almost directly. Almost directly it starts.
Right. He's right on the brink of doing a plaque assay.
Right on the brink. By the way, we can talk about 2 years later, there was a publication by a guy named de Herrell, who's often credited with most of the early phage work, and rightfully, because he actually developed a plaque assay,
right.
A quantitative plaque assay. Let's see. He says the condition or a disease could be transmitted to pure cultures for an indefinite number of generations. So he's got plaques. He can transmit it. It's filterable. He shows that it's heat sensitive, but if at room temperature, you can keep it for a long period of time. So he actually did a lot of very clever experiments with it. And then in the end, he says, not quite the end, he says, from these results, it's difficult to draw definite conclusions. What a letdown. In the first place, we do not know for certain the nature of an ultra-microscopic virus. And then he goes on about all of the different things it could be, and whether it could be living or not, and et cetera. And in the very end,
a question we still haven't settled.
Yeah, right. In the very end, says, in any case, whatever explanation is accepted, the possibility of its being an ultra-microscopic virus has not been definitely disproved, because we do not know for certain the nature of such a virus.
Right.
So cool. There you go. That's the first description of a bacterial virus, a bacteriophage.
Right. But then he ran out of funding.
Yes. The very last paragraph, it says, I regret that financial considerations have prevented my carrying these researches to a definite conclusion, but I have indicated the lines along which others, more fortunately situated, can proceed. Actually, it turns out he kept working.
Okay.
I had some correspondence. You dug up this.
Yes, the very interesting analysis later on. It turns out to be by a colleague of yours.
Yes, Donna Duckworth. And this we can post in the show notes, wrote this article, Who Discovered Bacteriophage? Because as I said, de Herelle, a Frenchman, came along 2 years later and described quite elegantly his research on bacteriophage. And in fact, he coined the term bacteriophage.
Which is why they're called phages rather than eaters.
Right. And he never mentioned twort. And so there's been some question as to whether or not he was aware of Twort's work. And it wasn't until 4 years after that, in 1921, that some other investigators, basically in trying to tell de Herelle that he was wrong, they weren't living things, they were an enzyme, dug up Twort's work and used that as evidence, because Twort was so sort of uncertain about what he had and was to some extent falling down on the notion that it was just an enzyme or something like that. But at any rate, these other workers kind of rediscovered Twort and brought it to de Herelle's attention. And so there's been some question as to whether de Herelle actually knew about Twort's work or not. And apparently there was a lot of stuff going on at the time as to who could really claim to have discovered phage. And Donna comes to the conclusion that these two guys discovered it independently.
Right.
I had some correspondence with Donna some time ago as I was reviewing this article again. I pointed out to her this bit about him running out of money. And she said he was always grousing about how he didn't get enough grant money.
Okay.
That sound familiar?
Yes, that comes up occasionally.
Yeah, that comes up occasionally. So,
yeah.
At any rate.
Yeah, so this, it was interesting to me that you see this just about every time there's a story about a scientific discovery, and so discovered this. Isaac Newton developed calculus, and they a little asterisk on that, and you read about Leibniz.
Right.
And de Herelle discovered bacteriophage. Oh, but then there was this earlier guy, Twort. Right. And it's almost universal that when there's a major technological or scientific innovation, it is not one guy who did this. It's an idea whose time had probably come, and usually you get a couple of different discoverers, often independently. And in this case, you're looking at 1915. World War I has not started yet, so there's still, has started in Europe, I guess, but hasn't gotten to full scale, and so there's still some science going on. The technology has come to the point where people can culture bacteria, they know about viruses, and this is where you're going to discover bacteriophage, and sure enough, a couple of different people did.
Right, and he wasn't really doing it in a vacuum. It's in the context of viruses and stuff. It's interesting to look at the title of this, An Investigation on the Nature of Ultramicroscopic Viruses. I think in that title, he's referring to his general investigation of trying to grow viruses, which basically was a failure. He was not able to do it. And he comes up with this other thing, which he's even reluctant to call a virus. He's not sure.
Right.
Okay. At any rate, there you go. Plaques.
Right. Because what he set out to do was just to understand how viruses worked. And he didn't accomplish that, and so he wasn't quite sure what to read into his conclusions.
At any rate, I really enjoy looking at these historical papers, and I think we ought to do some others at various times. De Herelle, of course, wrote entirely in French, so that we can't really do. Though I sent you a thing. Both of them, I guess, were invited to write sort of retrospectives for a journal called Science News in the late 40s. And those articles are sort of interesting. Though most of it is in Duckworth's, who discovered bacteriophage PDF, which we can't post because that's online. She has a nice summary of both of their discoveries.
Right.
So, okay, yeah, we ought to do some of these others, like in particular the Rouse paper. I'd like to do that.
Yeah, that's a pretty cool and very important event. So, cool.
Okay.
Neat stuff.
What do you got, Alan?
Well, the one I picked, we've got actually a big, long backlog of really cool papers, and then I went and picked yet another one.
Okay.
It just happened to come across my radar, and I thought, hey, that's a neat approach. So this is, it's a paper that came out in PNAS July 19th, and it's Michael Oldstone's group. Kevin Walsh is the senior author, and then a bunch of other authors on it. Suppression of cytokine storm with a sphingosine analog provides protection against pathogenic influenza virus. So what caught my attention about this is we've talked, whenever we talk about flu, we talk about the immune response to flu because it's such a huge part of the pathogenesis of that virus. And there's been a lot of discussion over the years that, if you could tamp down some of the immune response to influenza, you could probably save a lot of lives. Because when people get sick with influenza virus, they develop this lung disease, this pneumonia that is debilitating, but it's actually not the virus that's causing that. By the time you get to that point in the disease, the immune system is winning. But that's the problem. Because you've got all this inflammation going on, and you've ramped up this massive immune response that is just pounding the heck out of anything that looks like it might be the flu virus, which happens to include a lot of your own tissue. So you have this massive inflammation and something that's referred to as the cytokine storm, which is a feature that is not unique to flu. This comes up over and over again in pathogenesis of a bunch of different viruses and other diseases. So a cytokine storm is this immune response that gets out of hand.
Right.
It's you've got lots of inflammatory cytokines. Cytokines are the molecules that immune cells use to talk to each other and to coordinate their response.
Right.
And when these get above certain concentrations, and you get a whole bunch of them around, you've got this feedback loop that's self-reinforcing, and you get the inflammation running out of control. And that, in many cases, does more damage to the host than the original disease did.
Right. It's worth pointing out that the symptoms of most virus diseases are, in fact, not the virus, but the immune system.
Right.
Like rashes, blisters on the skin, even the flu-like symptoms that you experience with the malaise and fever and that kind of stuff, that's all immune response. And tinkering with that has always struck me as sort of a delicate business, okay? Because you do want to have an immune response, right?
Absolutely.
And so, my sort of knee-jerk reaction is, well, don't mess with that because you're just going to make the disease worse. But this paper has a different spin on that, which is interesting.
Yeah, and that's always been the problem. Is there's this number of years ago, people discovered that this cytokine storm effect was a major pathogenic mechanism, and it's why just about everything starts with flu-like symptoms.
Mm-hmm. Right.
You've got Lyme disease. It starts with flu-like symptoms. You've got influenza. Everything starts with flu-like symptoms because it's all this immune response. And people have known about this for a while and kept saying, well, maybe if we could tamp that down a little bit. But as you say, it's really dicey because when you start talking about suppressing the immune system, then, well, that's the problem in AIDS patients.
Right.
This is not something you want to toy with. But what this group did was they've been looking at this molecule AALR, which is a synthetic analog of sphingosine. And sphingosine is a signaling molecule that's involved in a bunch of different things, but one of the main roles of it is coordinating the immune response.
Right. So I want to spend just a minute on sphingosine here because this was new to me.
Yeah.
This is a lysophospholipid, right?
Yes.
So it's basically like a long chain fatty, well, not even that long, like 18 carbon, I think, fatty acid with a polar head group on it, but it's a single fatty acid chain. Most lipids have two fatty acid chains, and it is derived from, what is it, ceramide,
right?
Which serves both as a lipid and its own signaling molecule that has a lot to do with signaling, controlling apoptosis inside cells. And so there are receptors on cells for sphingosine or phosphorylated sphingosine in order for it to it has immune regulatory functions, as we will talk about. But I was unfamiliar with that as a signaling molecule. One of the things that interests me is lipid signaling molecules have the advantage that they are lipid soluble.
Right.
So they can get across membranes easily. And as a lyso lipid, that is a single carbon chain, it's also more water soluble than a regular lipid would be. So it can cross membranes and yet at the same time has some solubility in water and has pretty profound signaling activities.
Right. And even though it can cross membranes, there is a cell surface receptor for it. Mm-hmm. The sphingosine 1 receptor, or probably a couple of them. And this triggers cascades of events inside cells. It's the usual cell signaling story. But as I said, the immune system is one of the main users of this particular channel. I
couldn't find in the reading. I was kind of looking for some indication that sphingosine had more activity on one or another arm or aspect of the immune response, and I don't seem to be finding that. It seems to the effects that they're describing look pretty general to me.
Yes.
Is that your sense as well?
Yeah, I think it's kind of a blunt instrument.
Okay.
And which was a little surprising to me too. I immediately went and did background reading on sphingosine as well. And it's not quite as precise as you might expect if you're going after tamping down the cytokine storm, but
right.
That is the approach. And in an earlier paper, they took this AALR, the sphingosine analog, and tested it in mice that had been infected with a mouse-adapted flu virus. It's a mouse-adapted version of one of the human flu viruses. I think it was an H1N1. And they extensively characterized the immune response in these mice with or without AALR treatment. And they showed that this molecule seems to mitigate the inflammatory response in the cytokine storm. So in this paper, they're taking the next step. They're looking at all sorts of parameters, and they're also infecting the mice. They have a mouse model that they can actually infect with human H1N1.
Right.
And they're putting that in. They treat the mice. I thought this was actually a pretty good design overall. They've got one arm where the mice are treated with the AALR by itself. They've got another where they've treated them with oseltamivir or Tamiflu. And then they have a third where they've given combined therapy. And the importance of that is this is a drug that obviously they're hoping to get into some kind of clinical use, and you want to test your drug in the way that it might actually be used.
Right.
And of course, if you come out with a new therapy for flu, you have to assume that it's going to be given in combination with oseltamivir or one of the other antivirals.
Right, so it's appropriate to test them together in these preclinical studies.
Right, because if it turns out that that's lethal, then you got a problem.
Right.
So they find that in fact treating these mice, they looked at survival rates and then a whole bunch of pathology slides, and they found if you give the mice the Tamiflu alone, it helps, and compared to the controls that didn't get any treatment. If you give them the AALR alone, it helps a good bit more. And if you give them both treatments, they actually had, I think, a 96% survival, 27 out of 28 mice.
Yeah, I found these survival curves really pretty striking. So they've got they've got each group has 28 mice in it, so that's a pretty big group. And the controls that are infected but untreated, you're talking about 20% survival. And as you said, they got 96% survival with the combination of drug and oseltamivir. Though the drug by itself gives 82% survival.
Right.
So it's doing most of the heavy lifting.
Yeah, and oseltamivir only gave 50%.
Right. Right. So it's better than Tamivir by itself, and the combination of the two is even better. And it's really going from 21% to 96% is amazing.
Yeah.
Okay. Granted, it's mice.
Well, yes, that's always the caveat with these sorts of studies.
So what was it? Mice lie and monkeys exaggerate.
Yes.
And actually, let me go back for a minute to de Herrell, okay? Because I picked up a thing from his article. This is his article from 1949 where he's retrospecting his stuff. He says, I believe indeed that experimentation is significant only when it is carried out on animals naturally sensitive to an infection. This is the principle which Pasteur always observed, and its infringement inevitably leads to error.
Yes.
However,
but you got to start somewhere.
Got to start somewhere.
And
so there you go.
If you want to study a drug for human influenza, you can't just take it straight out of the lab and put it in people.
Stick it in humans. Well, you could maybe in 1915.
Yes, you probably could in 1915.
Okay.
So yeah, they got these great results off the top. I was a little surprised that they didn't have any uninfected control mice.
Yeah, right.
There are no mice that didn't get the virus.
In particular, for the pathology, it would have been interesting to see what an untreated lung looks like.
Exactly, and theoretically, you can just go to the whole library of slides of what a mouse lung looks like, but it'd be nice to have the same strain in the same experiment and all that, and right next to them. But the double treated line gives something pretty close to that, and it looks the lungs are healthy, and the untreated, the lungs are falling apart. And
there's a lot of infiltrative inflammatory cells.
Enormous amount of infiltrative cells, whereas in the treatment arms you see fewer, and then in the combined treatment arm you see very few.
Right.
So really, really positive results. And then critically, they looked at antibody production. Now, I say critically because the mainstay of combating influenza today is vaccination. So you wouldn't want a drug that hurts antibody production against the flu virus,
right? Because
that's kind of what you've gone for initially. And also that's obviously a key part of the immune response against the virus. And they found these mice, whether you treated them with Tamiflu or with AALR or with both, they produce antibody levels that are comparable to what you see in controls. So you're getting a robust B cell response, you're getting lots of antibodies, and that appears to be working.
Right.
It knocks the virus down, and they show some viral titer data, but there's a clump of data not shown that you pointed out. They don't
show the viral titers in animals treated with just oseltamivir or the combination, and I would have liked to see what that did.
Right.
But nevertheless, they show that it's interesting. The viral titers are about the same, whether or not they treat with the drug.
Right. With the AALR.
Right.
Right. Which strongly implies that this drug is not affecting viral replication.
Right.
So it's not affecting antibody production. It's not affecting viral replication, but it's reducing the cytokine storm and the inflammatory response and death.
Right. And then they measure, to back that up, they measure all sorts of different cytokines and chemokines.
Yes.
Interferon alpha, IL-6, TNF alpha, interferon gamma, CCL2, CCL3, CCL5, blah, blah, blah. And several others in different formats. And they show generally that the treatment with the drug does, in fact, tamp down the cytokine production.
Right.
And so it's working as they expect, yet at the same time, it's not really affecting. Actually, I guess the really significant thing about the virus titer data is that you don't get more virus in the drug-treated animals. Okay.
Yes.
Because you're tamping down the immune response. You might expect that under those circumstances, you would get more virus replication and cause a problem.
Right.
But what's significant to them is no, that does not happen.
Right.
Right? And you get it's interesting because even though you're tinkering like crazy with the cytokine response, that's apparently not sufficient to significantly affect the production of antibody.
Right.
So you got plenty of you got the antibody. Comparing the antibody titers, they don't change.
Yeah. So it seems to, despite pointing out that sphingosine's kind of a blunt instrument for this, it seems to have acted more or less the way you'd like it to. It has affected the inflammatory aspect of the immune response and the cytokines that are associated with that, but it hasn't affected the B cell response. It hasn't affected the ability of a virus to replicate. So it's not really messing with those other aspects.
So it would be really interesting to me to see how general this is with other viruses. In particular, other viruses that rely on one arm of the immune system or another, or where the either the cellular or the humoral response is more or less important in controlling a virus infection, and see to what extent this blunt instrument thing impacts different viruses. I mean, you pointed out to start with that the cytokine storm is a particular problem with influenza, and so this may not be as important for as impactful on other viruses where that's not as much of an issue. Or it could be that other viruses that interact more heavily with one arm of the immune system or another may be less or more susceptible to this as a drug.
Right.
So,
and it might be worth testing even against viruses where you don't think a cytokine storm is really involved. Right. Because you don't know exactly what you're hitting here, so it might be worth a shot.
I have this, I guess, naive idea that our immune response to something like an influenza virus infection is properly tuned, right?
Yes.
And it's going back to this thing where it doesn't seem to me like tinkering with that is a good idea. But this whole conversation suggests that it's not necessarily properly tuned. It can get, especially in the extreme cases, out of balance. You're kind of overdoing it in your immune response to the point where you're doing yourself harm. And if you can use a drug to back off on that a little bit, there actually may be an advantage.
Yeah, and I think it's important to remember that the evolution of the immune system wasn't based on kind of a medical concept. It was based more on a public health concept. Right. So the important thing for evolution is that the species survives. Mm-hmm. And if a few individuals develop allergies and anaphylactic reactions that kill them, but the upside of that is that you get a robust immune response in the rest of the population that saves their lives, then that's a worthwhile evolutionary trade-off. Right. Now, in modern times, the individuals who get those allergic reactions and anaphylaxis and autoimmune diseases would really like those to be treated.
Right. And they're the ones that wind up in the hospital.
They're the ones that wind up in the hospital. They're the ones who you need to treat with something. Right. So in a case like this, you have influenza. And the truth is, as much as we've talked about the deadliness of flu, the majority of people who get it do survive. Mm-hmm. It's just that you don't want to be rolling those dice personally.
Right.
So it has not wiped out the human species because the majority of us can survive long enough to reproduce, even in the presence of influenza. But if you're one of the unlucky ones, you'd really like to have a treatment that can tamp down this cytokine storm.
I guess the other thing is that evolution isn't done, right?
Yes.
We are at a certain point in this game, and there's a tug of war going on between our immune systems and the viruses. We're not finished yet.
Right, and our evolution is not done, and the viral evolution is not done.
Right.
And the viral evolution, I kind of doubt that there were global pandemics of influenza in the time before human cultures all had contact with each other.
Right.
So one of the things that happened when Europeans came to the Americas was people started dying of diseases that they never had before.
Right.
Now we get these global pandemics every year because we have this interconnected world. So we've toyed with the virus's evolution, and now we need to toy with our own evolution as well.
Well, I wonder how close this drug is to any sort of clinical trial. This is just mice. I mean, you're going to have to probably have a non-human primate thing going on.
Right. There'll at least, I think, in flu, probably the next thing to do would be ferrets, right?
Yeah. Okay.
Or maybe, I'm not sure. Maybe ferrets or.
And there is a relative of this drug that's actually FDA approved, right?
Yes.
Here it is. Fingolimod.
Right.
And I forget what that's approved for. Do you remember?
I do not, and I'm.
All right, here we go. Wiki.
Yeah.
Approved for treating multiple sclerosis.
Yes, that's right. So I'm not sure exactly what the mechanism there is in MS, but
I'm not sure they know.
Yeah, that's a lot of drugs are that way.
Oh, yeah. Here it says, it can sequester lymphocytes in lymph nodes, preventing them from moving to the central nervous system for autoimmune responses in multiple sclerosis. Of course, right. MS is an autoimmune disease.
Right.
Okay. So if you tamp down the immune response, then you got to treat it. Now, I don't know if this particular drug has the same sort of activity as this other one, but this is a situation where if it's approved for something else,
it could be used off-label.
Right.
Now, I don't know if that's been tried, but usually off-label, it's difficult to collect data.
Right.
So it seems like somebody ought to set up a clinical trial of the approved drug.
Right.
Or there may have been a rationale. I looked around a little bit for the rationale for using this AALR versus the fingolimod. Why not do your experiments with the approved drug? Mm-hmm. But I didn't come up with anything. So I assume that Oldstone's group had some reason for using this other molecule.
All right.
But yeah, the next steps would be trials probably in a couple of different animal systems. Usually, you'd like not only mice, but maybe some other rodent system, or perhaps ferrets, and also go into non-human primates, and then test in a phase 1, make sure it's safe in humans, and then start looking for efficacy.
10 years, 15, something like that.
Maybe 10. It might be accelerated a little bit for flu.
Right.
Because you could get some data from, I don't know, maybe emergency use, depending on how pandemics go. But we'll see. It's certainly a promising approach. And
I think it's really interesting. And as I said, that although it's mice, the initial that first figure was showing the survival in the mice is really dramatic. And everything else is consistent with it. It makes perfect sense, and it's very instructive about sort of the nature of the immune response. It'll be interesting to see what happens with this.
Yeah, and I think it's cool that we're almost to the point where we can second guess the immune system and maybe do better. And I say almost and maybe.
Right.
But that would put us, just a little bit behind the viruses.
Right.
Which can second guess the immune system consistently.
Yeah, well, they've had 4 billion years.
Yeah,
to
they've had a little while to work on it.
Right.
So.
Okay.
All right.
Excellent.
So I think it's about time to move on to some letters, which we have a few of. Yeah.
You want to take turns doing this?
Yeah, let's go ahead and do that. Do you want to take the first one?
Sure. David writes, this is David, who is he's a professor in the biology department at Vassar College, and he has written to us on a number of occasions.
Yep.
Hi, TWiV folks. I am developing a class on how infections can alter the behavior of their hosts. There are some fantastic examples of this in parasitology, where the parasite alters the behavior of the host to increase its chances of transmission. The reason I decided to develop this class is that I find it fascinating that rabies induces aggression, causing the infected animal to bite and transmit the disease. But I don't know anything about how it actually does it. I'm developing the class with a physiological psychologist who does animal behavior research, and we are already having too much fun learning about zombie ants and other strange infections. My question is, other than rabies, are there other viruses that alter host behavior, especially with the effect of increasing transmission? It seems that there must be a sexually transmitted disease that increases sexual desire or promiscuity, but I haven't found one. Do you know of any? Also, do you have any thoughts on the origins of the mythology of zombies and vampires as possibly based on behavior mediating infections? Certainly, many zombie movies portray zombieism as an infectious disease. Could these ideas have started with historical cases of altered behavior due to infection? Cheers, David. Well, he's way out of my league with this one.
I think well, first of all, that sounds like an awesome class.
It sounds like a fun class.
I would totally take that course.
Yeah, and if he's got some students who are engaged, they'll go out and they'll find their own stuff,
yes,
and bring it back to him.
Yes, they will. They will teach him. My thinking, I had always just assumed that the zombie concept probably came from rabies, if it came from anything, because that's the most, that's the most obvious parallel.
Actually, did you look this up?
No.
Did you look up zombies?
No, I did not. In my research for the show, I failed to look up zombies. I'm terribly sorry.
Yeah, let me see. We got Wiki here.
Well, there's a history of this in voodoo,
right?
With I think it's powdered pufferfish that's used as the toxin to zombify somebody. But I know let's see.
I can't get a lot of history out of this. So I don't see anything about rabies.
Yeah, it's derived from a Haitian Creole word, actually.
Uh-huh.
An animated corpse brought back to life.
Okay. So, and I don't know of any examples of, and we actually, tell you the truth, I encountered David at the ASV meeting.
Oh, okay.
And we had some talk about this, and I think we had talked with Vincent about it as well. I can't think of any sexually transmitted diseases where the infection increases sexual desire. That would be marketable.
That would be extremely intensely studied, because
Right.
if there were a button you could push in people that would have that effect.
There's a lot of stuff that's sold under that general concept.
Yes,
But, I can't think of any infectious diseases.
No, I don't think so.
There's another. This is not virology, but one was brought to my attention. One of these sort of relationships recently. This was brought to my attention. There is a moth that has a behavior that allows him to avoid bats. The moth can hear, in quotes, well, I guess it's not even in quotes, the moth can hear the bat sonar and reacts by basically folding up his wings and diving.
And falling, yeah.
And evades the bat. There is a mite that infects the ears of these moths, okay? But in any given moth, the mite will only colonize one ear,
okay, leaving the other ear available so that it can hear the bat. Otherwise, it would be to the detriment of the mite.
Right.
And it doesn't matter which ear it is, okay? And that's random, okay? And the way apparently this works is that once the mite lays eggs in one ear, there's some sort of pheromone or chemical signal that goes on so that any subsequent mites will colonize the same ear and they don't go to the other ear.
Right.
Cool. So that's not virology, but it's another little tidbit.
Yeah, that's kind of an example of a pathogen, engineering, and not behavior, but physiology, I guess.
Right. Well,
but the other thing to remember is myths and things like that don't necessarily have to come from a specific natural phenomenon.
No.
As long as they're cool stories, or if
Right.
they served some religious purpose, then that's what gets these types of things perpetuated.
Right. Okay.
All right. Now, Wendy writes, I was recently listening to your 100th podcast where you featured David Baltimore. One aspect of virological history that was discussed but is frequently overlooked is the work by several investigators at NIH in the late 1970s to determine if indeed cloning was safe. These investigators took time from their own studies to identify dangers of cloning and whether such studies could safely be performed. The studies were very important in the history of molecular biology as well as virology, and I believe that the details of these studies would be of interest to the community at large. Those individuals who did this work are getting older, and that history will be lost once they're gone, since there seems to be few details of this work published, as far as I can tell. How about getting some of those scientists on your program to talk about the events? And Wendy's at the University of Iowa.
Great idea.
That's an excellent idea.
I think, Vincent at least was, has some knowledge of this. He wasn't involved in the conferences. The conferences that would happen were the famous Asilomar conferences.
Right.
So when people figured out that they could clone genes, some scientists sort of said, hold the phone. Maybe there could be bad consequences of this. And it's really, much to their credit, they imposed a self-moratorium on the work.
And to put this in the historical context, this is happening at a time when the Cold War is raging.
Right.
Which is, I think, important. Where you're dealing with people who grew up in the generation of science having produced this incredibly deadly and problematic technology during World War II, and now the biologists have discovered something that might or might not be in a similar category.
Right. And so they actually got together and they had a conference on the West Coast near Big Sur at Asilomar, with all of the major players involved, and kind of hashed it out and came up with some concepts and some rules. I frankly don't know the details. Vincent might know more, because Vincent was at the time on the verge of cloning the polio genome, right? And they were all set to go, but there was a self-imposed moratorium on the on recombinant DNA, and it wasn't until that was lifted that they went ahead. Now, politics crept into this eventually too, because it wasn't all self-imposed. In Cambridge, there was actually a local government moratorium on recombinant DNA experiments, and there were some broader prohibitions nationally in the U.S. I remember people going to abroad to clone stuff because you couldn't do it in the U.S. This was once some of this stuff sort of settled out. At any rate, I think Wendy's right. It would be interesting to get somebody on who was at Asilomar. Well, we missed an opportunity with David. I'm sure he was there.
Well, we had a lot of stuff to talk with David about.
That's true. That's true. But let's get somebody on who was there and can talk about it. It's a very good idea.
Yeah, definitely.
All right. Joe writes, "Dear TWiV cats."
I like that.
I would like to wade into the viruses aren't they alive debate with a thought experiment.
Like every person on the planet, I am related to every other terrestrial organism via a common ancestor. For a chimpanzee, our shared relative lived relatively recently, geologically speaking, whereas for E. coli it was probably billions of years ago. However, we share grandparents and are all very distant cousins. So, with this in mind, am I also related to poliovirus? Is Ebola my tearaway cousin, 10 to the power 30 times removed? And if we are related to viruses, does this have implications in the alive, not alive, undead status? Just a thought. Love the show. Thanks, Joe. Joe is from London.
Interesting.
Wow. Yeah, interesting.
That's an angle I hadn't thought about.
No, I hadn't thought about that one either.
Well, pox viruses certainly have some borrowed genes in them, or something.
I mean, basically, this goes. I would say if he's going to say like, well, first of all, I'm in the viruses are alive camp, so I don't have much problem.
Yeah.
With this, okay, and I'm sort of into the whole Gaia thing. So if we're all related to every other terrestrial organism, yeah, sure, I'm related to viruses. I got nucleic acids. They got nucleic acids. We use the same genetic code, right?
Yep.
Okay. So as far as I'm concerned, sure. Me and Ebola are from the same lineage as is poliovirus. I don't have any problem with that at all.
Yeah, now it's too bad Vincent's not here because he's against viruses being alive. I'm actually leaning toward viruses as living.
It's obviously a complicated debate because if, as we said before, if viruses are living, then maybe transposons are living as well.
Right.
So it gets a little complicated. But from my point of view, I don't have any problem thinking that I'm related to viruses. As a matter of fact, that's kind of a nice thought.
Yeah,
I like that.
Yeah.
That's pretty good. And it's all wrapped up in the yes, alive or not alive status. If I'm related to viruses, then yeah, we're both alive. That impacts on that.
Yeah, and conversely, if you're both alive, then you're related.
I like that he managed to slip undead in here.
Yes.
We seem to have a zombie theme in this episode.
That's always an important category to include.
Well, Joe, thanks for your thoughts. You're really you're with us on this. You're thinking way into it.
Yes.
That's good.
All right.
This next is definitely for you.
Yes, a different Joe. This is in regard to agave viruses, also responding to Kevin's letter on TWiV 137. As a longtime listener to TWiV, I was dismayed that the crew was not familiar with the groundbreaking work of Professor Jose Cuervo regarding the agave life cycle. He identified the TWiV virus as its primary infective agent and showed how it was transmitted by the common barfly. It's one of the few agents that also causes diseases in humans, the dreaded morning after sickness, with the classic symptoms of headache, dry mouth, blurry vision, and tremors. Love the shows, Joe. And Joe is at LSG. I'm not sure what LSG is. P.S. Alan can probably come up with a better virus name. Feel free to edit. No, I think I'll leave TWiV virus, but we probably should have riffed on that a little bit. I think Dr. Cuervo actually collaborated with the Scottish researcher John Walker, and we could also have mentioned the very, very important studies by Dr. E.J. Gallo on the Vino Veritas agent, which I believe is closely related. And VVA has been implicated in drunk dialing syndrome, which back in college I was once a sufferer of, and it's not a pretty condition. So.
Is that right?
Yeah.
Well, Joe has too much time on his hands, I think. But I really appreciate this. It's nice to have a little humor from our listeners. That's good. Jeffrey writes, hello, in TWiV 139, after describing the loss of the fiber genes from Mimi virus genome upon passage, one of you asked whether if other viruses lose their fibers as well, and someone mentioned adenoviruses. I am now a community college assistant professor, but in my former life, I was a researcher and did my dissertation on the persistence of adenoviruses in human lymphocytes. Bringing up adenovirus was interesting for a couple of reasons. First of all, if you compare the genomes of the animal adenoviruses, the conserved genes are in the middle of the genome, and its ends are the most different. And Jeffrey cites a reference for this. Second, its fiber genes are in the middle, not at the ends, and appear to be pretty well conserved. Anyhow, love the show. Thanks for keeping me current on research now that I have to spend my time grading. So I found this actually very interesting because one of the tenets of poxvirus genomics for 30 years has been the observation that in poxvirus genomes, the genes that are in the center of the genome are the highly conserved genes, and out towards the end are the genes that are less conserved. And the thinking has always been, and this is borne out by years of experimentation, is that the genes that are in the middle, being the conserved genes, are the nuts and bolts, the housekeeping of replication, polymerases and structural proteins and stuff like that, and that the genes out on the end that are more variable are responsible for the individual characteristics of the virus, like host range and pathogenicity and stuff. So that's where immune modulatory genes hang out and that kind of stuff. I was not aware that this apparently is also a feature of adenoviruses, and if you look at this, it's a review that he sent us. It's also herpes viruses as well. So
and these are not closely related viral families.
No, so it
seems almost like this is a general. I looked at that too, and I thought that's interesting. That's almost a general theme of linear DNA viruses, right?
Right, and there was some sort of comment in that same article about there being this. I forget exactly, but it seems to hold up in yeast as well, as if there's more variability near the telomeres in yeast. So I don't know how that would work mechanistically. Maybe maybe this has something to do with recombination and exchange of information, that you could get away with doing this at the ends without trashing the conserved stuff. There must be a mechanistic reason why the conserved regions would be clustered in
a selective pressure.
Center. Yeah.
It could be something like ends are more likely to be damaged.
Something like that.
So you would naturally select for organisms that pack their most important genes towards the middle of chromosomes.
Right.
And then evolution would take care of the rest.
Right. So that's very interesting, and I appreciate him sending that in. And yeah, I would have thought the fibers on adenovirus, they have a very specific function that's critical for getting the virus into the cell. So it doesn't surprise me that they're conserved, and I would be very surprised if you could lose them without consequence.
Right.
So.
Yeah, neat pattern though.
Yeah.
And thank you, Jeffrey. That's
yeah.
We wouldn't have come across that otherwise, probably.
Okay, so let's do this one.
Yeah, so we were going to save this last one for Dixon for when he's on the show.
But Dixon spends too much time fishing.
Who knows when he's going to be on the show again?
Right.
So we know Vince will be back, but Dixon, he'll one of these days. So we'll go ahead and read this one. This is from Jeffrey. He says, I feel that I must rebut a point made by Alan Dove in episode 130. He quotes me, "We're not looking at a world where we have a choice between these genetically modified pest-resistant plants and some beautiful organically grown, no pesticide, no evil chemical, nothing types of foods. In fact, with more than 6 billion people on the planet, what we have is a choice between these pest-resistant genetically modified plants and plants that are grown with petrochemical pesticides." And I do recall saying something more or less like that in response to somebody else's letter. Jeffrey says, "First, ask Dr. Dupommier to describe permaculture to you. It's proving to be an increasingly more viable method of growing food while improving the environment. Farmers like Mark Shepard in Wisconsin," and he gives a couple of websites, "are showing that permaculture can be financially viable and product ton per acre efficient, a way to produce food without pesticides. Second, there's at least one other alternative to grow enough food to feed our planet. Grow your crops within a semi-customized ecosystem rather than in isolation, and direct natural biological controls toward keeping your crops happy as a first line of defense. Use pesticides as a highly selective and time-limited second line. This is not to protest against genetic engineering. I strongly believe that it has a valuable place in the future of our culture, but it should be used in a manner that promotes rather than degrades the health of the planet. Yes, this type of agriculture requires a complete restructuring of our current agricultural system, but then what do we think will happen to the system when gasoline and related petrochemicals reach $5 or $10 a gallon? Thanks for the excellent show.
So first, can you describe permaculture?
Right. So we touched on this once a little while ago, I think, when Dixon was on the show. Permaculture is essentially the mashup between landscaping and agriculture. And this is the idea that instead of just taking your flat field and plowing it and planting your monoculture of corn in it and doing what you have to grow as much corn as possible per acre, you actually maintain a mixed ecosystem, essentially. It can also include sections of forest. It can include a variety of different types of plants. And you shape it in such a way that it's permanently maintained to yield food for people, but also to function as an ecosystem for the environment that it's located in.
So with respect to the issue of pesticides, I guess the idea is that if you take one crop and stick a whole ton of it out in one place, in terms of pests, you're just asking for trouble.
Yes, because you've set out a banquet.
Right.
Whereas if you've got the crops scattered around, I can give a very, very small example of this from my own extremely limited gardening experience. There's this notion that you plant carrots, I think it's carrots and onions, in between each other. Because it turns out there's a fly that parasitizes carrot plants. And there's another fly that parasitizes onion plants, and they don't tell the two apart very well. So if you've mixed these two in between, then the flies will often try to parasitize the wrong plant, and it won't get anywhere. And it's kind of a pest control situation. Now, if you magnify that type of idea up to a whole landscape where you're doing little tricks like that all over the place, and you've got your crops scattered out, it's very difficult for any pest or disease to get a major foothold. And you also have all the predators that would be there. You have spiders eating the bugs. You have birds coming in and eating things. So you kind of integrate the farm into the ecosystem. It's a really, really cool idea.
I'm also reminded of our conversation with Marilyn Russink, where she talked about the symbiosis that goes on between viruses and fungi and plants in a natural setting, whereas the cultured plants don't have the same symbiosis going on and are less healthy as a result.
Right.
Okay.
Right. So it's I before I give my answer to this letter, I want to point out I'm pro permaculture. I think it's a great idea. It's a neat approach. However, if updated figures now of 7 billion people, which is what we're forecast to get pretty soon, show up at permaculture's front door demanding breakfast, the answer is going to be, we're not quite ready for all of you yet. Because permaculture is a technology that some people are trying, and this Shepherd's Farm in Wisconsin is a good example. It's currently operating at small scale in a few different places, but it cannot feed the planet right now.
Right.
And my point was, right now, we've got all these mouths to feed. And you have to keep doing that while these beta release technologies, and permaculture is one of them. There's also a slightly less revolutionary technique called integrated pest management, which I think Jeffrey's hinting at actually in this as well. And it's kind of a similar idea. You use less pesticide and you end up getting a reasonable compromise. And maybe even further down the road, something like vertical farms that Dixon's been promoting. We're waiting for those things to get up to speed, and I think we're waiting to figure out how that's all going to shake out. And yeah, we have to restructure our entire global food infrastructure, which is to say our infrastructure for everything, to accommodate some of these changes. In the meantime, we've got to still keep feeding the world. And my point was, it's that intervening period of time where you really are choosing between genetically modified plants with Bt genes to resist insects or petrochemical pesticides.
Right, and you hope that somewhere in the future all that stuff will become obsolete, but it's an interim solution.
Exactly. So next spring's planting season is not going to be permaculture. It can't be. We can't get it done that fast.
This reminds me of conversations I used to have with my father. He was an organic chemist who spent his career working for the Chevron Research Corporation, which is the research arm of the Standard Oil Company.
Right.
And he used to have issues with people who basically wanted to immediately transfer everything to solar and wind and alternate sources of power. And his rap was always, yeah, fine, but you just don't understand the magnitude of the problem.
Yeah.
It's just absolutely huge. And yeah, we should be looking for alternate sources of fuel, but you really need to appreciate just how big the problem is.
Yeah, and if you go out to the Midwest and you see a mile-long train of hopper cars full of corn. Now, there's a lot that's wrong with this picture, but you've got to replace that mile-long train full of corn right now. How are you going to do that? It's just the magnitude of it. You can't do it right now with these technologies as they are. But yes, further down the road, 10, 20 years from now, it would be great to move toward that. Just in the meantime, we need solutions that'll work.
Cool. Well, I wish us luck.
Yes, me too. All right, so shall we do a couple of picks of the week?
Sure. You first?
Oh, okay. So mine is more technology than science, but it's got some scientific angles to it as well. It's the AT&T Tech Channel. I stumbled on this a little while ago. I don't even remember who sent it to me, but these are documentaries shot by AT&T going way, way back. Some of them are training films that they used to show to their technicians. Some of them are documentaries promoting different technologies. And it's ranging from the new technology of solar cells to ballistic missiles and how they work, and very 1950s version of cartoons of how these things work, presumably to defeat the Ruskies. And then you've got things like a training film on how a telephone trunk line works.
Wow, this is cool.
Yeah, and it's just a neat vintage technology type of archive, and they're all
I like the old guys. Here's a 1970. This isn't that old, but here's a William Shatner.
Yes,
showing us how microprocessors work.
Yes.
Wow, from 1976, but they got stuff going back to 1938, 1930.
Yep.
Wow, this is great. I'd have a look at some of these.
Yeah, so that's fun stuff.
All right. So let's see. What do I got? Oh yeah. So. As I was researching the TWiV article, I needed to look up some stuff about vaccinia because I couldn't. TWiV was trying to. One of the things he was trying to do was to grow vaccinia,
right,
independent on its own. And this is where I say I have trouble getting into his head. Okay, like
what were you thinking,
dude? It's a virus. Don't you know? Okay, and so one of the things I looked up and found that in fact it was known in TWiV's time that it was a filterable agent. Okay, and a matter of fact, Negri showed that. And at any rate, so they had quite a bit of information about it, but still, I guess TWiV wasn't convinced that you couldn't grow it by itself, and I guess that wasn't unreasonable. Where I got that information, this is a long introduction, where I got that information was from a book called Smallpox and Its Eradication by Frank Fenner, who we've talked about before, D.A. Henderson, who we've talked about before, and Arita Jezek and Ladny, published by the World Health Organization in 1988. And this is a 1,400-page book that there were very few copies printed, I think. I actually have one on my shelf. Every credible pox virologist really needs to have one of these, so you can't get one anymore. And since they're out of print, they put it online. This has an interesting history. This book
and by the way, thanks for linking to a 1,400-page PDF.
Oh, sure. You're welcome. Actually, they've done it nicely because it does have a table of contents, so you don't have to load the whole thing. One of the mandates of the smallpox eradication program by the World Health Organization was not only eradicate disease, but then document what you did. Okay? And so this is the documentation. Written, Frank Fenner just was one of these guys who just had to be writing all the time. Okay? So I think most of this is actually written by Fenner himself. And it is a wonderful description of the pox viruses, the history of vaccination, the history of smallpox, and exactly the bulk of the book is exactly how the smallpox eradication campaign was done, country by country, and it's illustrated with lots of pictures and anecdotes and etc. So it's a fabulous resource for anything to do with pox viruses or smallpox, and it's a wonderful history of the eradication campaign. So,
not a cover-to-cover beach read, though.
No, not at all.
But a great reference.
It is a reference book that I use frequently.
And great that it's online.
It's great that it's online. As a matter of fact, this is what reconnected Vincent and me some years ago, or a couple of years ago, because he was writing a blog and had a pox question. I hadn't spoken with him in probably years. Okay, and he emailed me a question about pox viruses, and I immediately went to this resource and had the answer for him. And here we are doing TWiV without Vincent. Okay. So we got a listener pick of the week?
Yes, we do. So this is from Asif, who is in the School of Medicine at the University of Tokyo. And he sends this site, which I am going to find extremely useful.
Oh man, this is unbelievable!
And even if I don't immediately use it for my work, it's just awesome to browse. It's called Science Photo, and this is a UK a photography rights management company. They're like Getty or any of these other image shops. But they specialize in photos related to science.
Apparently, they've been around for quite a while.
Yeah, and they also have videos.
Oh, I didn't catch that.
Yeah, so you can go to the
motion.
Yeah, the motion section,
okay.
And you can load videos that they're available. You can just watch them, of course, and that's what most people will probably do. But if you want to use these for some purpose, you can contact them and license them. They're all available for licensing. Now, I don't know what the prices are or the terms, but the workmanship in these is outstanding. The video quality is great. The image quality is great. And some of the images are just really, really stunning.
I have to admit that this is a little morbid, but they have... Did you look at public health images?
Yes, I did. Yes, I went right there.
It's incredible. They've got the one I like is this. They've got a CT scan of an intensive care patient, and what you see is this skull and all of this instrumentation around it. He's got a tubes going in and coming out, and neck braces and all of this other kind of stuff. Amazing.
Yeah.
And the hand of a corpse.
Hand of a corpse are really, really haunting image.
God. Yeah, it's very haunting images. But this is a great site. I really appreciate it.
Yeah, and they also have these some of these other images are sort of creative design efforts, conceptual images that could be used for magazine covers or something. Those are kind of cool too.
Very good. And Asif is from Tokyo.
Yes.
Thank you very much. That's terrific.
Thank you. Awesome.
Well, I guess that about does it. If you like TWiV, you might also like TWiM, This Week in Microbiology, and TWiP, This Week in Parasitology, also Vincent's Brain Children. You can find us on Facebook. Just search for This Week in Virology. We have a sort of a fan page there. If you sign up, that gives us a little boost. The shows are available at iTunes or at the Zune Marketplace. Consider subscribing. It's free and allows you to automatically receive each new episode as it's posted. You can also listen with the Microbe World app. It streams episodes to your iPhone or your iPad. You can find that at microbeworld.org, and it's now also available for Android devices. You can also just play or download episodes directly from our website at TWiV.tv, where we also have the show notes. All of the links that we talk about and everything else are posted there on the website. If you like TWiV, please tell others about it. Links and reviews in iTunes are particularly helpful. They help us keep us on the front page. Obviously, we love to have email, so any questions or any suggestions of what you'd like to hear, you can email us at twiv@twiv.tv, or check out our new phone line, 908-312-0760, and you can leave a message for us there, or you can post at facebook.com This Week in Virology. So, thanks, Alan.
Thank you.
This was good fun.
Always a pleasure.
Great fun.
Yeah.
We've, I think we made it. I think we did it. We got all the way through without TWiV. We're not going to have another TWiV next week. Vincent will still be on the road, but the week after that, we'll have another episode.
Yeah.
So that about does it, huh?
Yes, indeed.
You've been listening to This Week in Virology. Thanks for joining us. We'll be back, not next week, but the week after. Another TWiV is viral.